Okay, for question 4A, I was very generous with the marking. Basically, you could have said point source or non-point source. I'm not sure what ID we're looking for here. If we see a guy with a little backpack and a nozzle spraying the field with pesticides, and then we find pesticides in our water, and perhaps we're at a farm living nearby, then that's going to be point source. In other words, we know where the pesticide is coming from. So if you mention like an individual or a farmer that's spraying pesticides, and that therefore it's an example of a point source, then I gave it correct for 2 marks. On the other hand, if we're looking at a large area and we've got lots and lots of monoculture, perhaps like lots of cornfields, and all the farmers have got sort of big sprayers, and maybe there's an aeroplane that comes and sprays, does crop dusting and sprays the crops. We don't know which particular place the pesticides in the water, so when we test the water, perhaps it has pesticides in there, we don't know whether it's come from this farm or it's come from maybe like a fruit orchard that's further up the mountain that also uses pesticides, so we can't be certain. So all of this water is running off into the watershed, and then we find it in our water, in which case it's non-point source. And so if you describe that kind of scenario with the runoff and the pesticides being applied in a broad spectrum, then I gave you the 2 marks for that. So now we have a table that shows us the relative abundance of primary consumers, birds, and secondary consumer birds. So the primary consumer birds, they're feeding on perhaps fruits, perhaps nuts, and the secondary consumers are feeding on insects most likely. So why have the secondary consumers been more affected? The primary consumer numbers have stayed about the same. So one possibility that you can talk about is that the prey for the birds has declined in numbers because of the pesticide coming in and killing the insects. So because insect population has declined. So they would be affected more than the primary consumers who are eating the fruits and berries. Also, the prey for the birds, these birds are feeding on insects which have the toxins in them. So they accumulate, so this is this concept of actually biomagnify, they magnify the effect of the pesticide. So these insects here are being eaten, and perhaps this insect is being affected a little bit, but as it goes up the food chain, okay, the birds are higher up the food chain, they're going to be accumulating that toxin, and it's going to get concentrated because there's a number of insects that they have to eat. Now, the plant-eating birds, or the fruit and nut-eating birds, they might also get a dose of the pesticide, right? So there's particles of pesticide, sitting on leaves, sitting around in the environment. They are going to get some of that, but it's not going to be as concentrated as in the for the secondary consumers. Okay, you needed to explain these 2 factors here to get 2 marks, or you could explain one of them. So, for example, this one about biomagnification, being a secondary consumer in detail, and then that would get you the 2 marks. Okay, for part C, they're asking about a biotic index. And some of you have completely forgotten what this is, but some of you vaguely remember. However, a biotic index is not about the abundance of a species. Okay, it is not about the abundance of a species. And that's because we might very polluted. Let's imagine like this swampy, oily, horrible patch of water that's got lots of yucky pollutants going into it. And we might find hundreds of little mosquito larvae, thousands of them, and maybe like little bloodworms, and other species that are highly tolerant of pollution. So we might come back and we might find out that for mosquitoes, we collected 1,000 larvae, and bloodworms, we collected 3,762. So actually, our abundance could be quite high. But what scientists know is that the presence of these organisms is not so much an indicator in itself of poor quality water, but that they are the only things that can survive in poor quality water. Now if we go to an area, perhaps a really pristine stream or pond, lots of biodiversity, very, very low pollution. Now when I do my sampling, perhaps I find, 15 different kinds of species. And perhaps one of those species is like a mayfly. And one of those species is damselfly. And these are much more sensitive to pollution. So if they're present in this pond, then we know that this pond cannot be very polluted. Now I might get, let's say I come back with 72 mayfly and 34 damselfly, and my numbers are all similar, so that my abundance down here comes to something like 1,064 individuals. The abundance is still less than over here with my mosquitoes and bloodworms and my swampy, polluted, toxic waste over here. But it's not to do with the abundance, it's to do with which species are present. These species are sometimes given points so that you could add up. Maybe this is a 5.1 and this is 5 points, or 72 at 5 points, whereas the mosquitoes and bloodworms are only 1 point. So you end up with an index at the end. You don't just have to do it in water. The other thing, some people did mention it, they mentioned plants, but they're actually lichens that grow on trees, and they're very sensitive to pollution. Some, again, are quite generalist, like our insects over here, but many of them are specialized and very, very sensitive to pollution, so they just disappear when there's a lot of air pollution around. Okay, for part 3, clearly nobody remembered what you did on the barge only a few months ago. So for this one, the biotic index, you're going to go and collect some samples of the water. And you can do that in a variety of ways, but why not describe the way that you did it on the barge? You collected the plants and all their roots, okay? And then you surveyed for invertebrates. So this would be 1 mark. You identify. If you don't know which invertebrates you have, you can't use a biotic index. So maybe using a key or comparing to pictures. You compare them to a standard. So in the past, scientists have established that certain species are going to be very sensitive to pollution and others are very resistant to it. So by comparing to those known values, you can know how polluted your water is. Of course, you don't just want to take your sample from just one site in the river. You want to take it from a variety of locations, and perhaps you want to compare it to an area that's not polluted in the river. So you might take several samples from an area upstream of the pollution, and then several samples in the area that is polluted. So these 4 aspects are really the ways to get the 4 marks. You need to collect multiple samples throughout the study site. You need to survey for invertebrates and talk about how you're actually going to collect the invertebrates. You don't have to do it with the plant. You could do it with a dip net, but in other ways. And then identify and then compare to the known standards. Finally, name one physical attribute of the river that could be measured as a part of assessment of this pollution. So, here you can talk about any other aspects of your pollution. So you've got phosphates and nitrates are the most obvious ones. You want to know if your water is polluted. Turbidity is a good one, the amount of particulate matter. pH is okay, but it's only going to really show up if there's really extreme things happening, and then you probably wouldn't find anything, but I awarded you a mark anyway. Phosphates, nitrates, you could do heavy metal testing, so for like mercury and lead. Fecal coliform, that was another one they talked about on the barge. So all of this stuff came straight out of your barge trip. So I think most of you got this mark and almost none of you got this one about biotic index.